Subsequent to the recording of this interview, telosatuzumab vedotin received accelerated FDA approval on May 14, 2025 for the treatment of adults with locally advanced or metastatic non-squamous non-small cell lung cancer with high c-Met protein overexpression, greater or equal to 50% of tumor cells with strong 3+ staining, as determined by an FDA-approved test and who have received a prior systemic therapy.
Hello, I'm Joel Neal. I'm at Stanford University, director of the Thoracic Oncology program here and a thoracic medical oncologist. Today, it's my pleasure to share with you updates in some of the targeted therapies for non-small cell lung cancer, including ERBB2/HER2, MET alterations in lung cancer, BRAF mutations, and some emerging data in KRAS mutant non-small cell lung cancer. Here's an overview of our agenda for today. I won't dwell on this, but provide it for reference. We'll be going through all of these papers and dive in a little more detail on some of these that are new and emerging. First of all, we'll start off with ERBB2 mutations, also known as HER2 mutations in non-small cell lung cancer. There's a variety of different mutations that we observe, including most commonly, as you can see in the red box, the exon 20 insertion mutations. The most common of these is the 772 duplication YVMA. You can see that makes up almost a third of observed non-small cell lung cancer with exon 20 insertions, but there's a variety of other mutations which can be activating, including the point mutation over there on the far left, S310F. As we think about developing targeted therapies for those, some of the targeted therapies that we have work in an extracellular way on the outside of the cell, particularly antibody drug conjugates. Others are more classical tyrosine kinase inhibitors that work on the tyrosine kinase domain. So we'll be talking about both of those today. First, we'll talk about T-DXd, also known as trastuzumab deruxtecan, for HER2 mutant non-small cell lung cancer. So we'll be talking about both HER2 overexpressed protein expression and HER2 mutant. But first, starting off on the established data for HER2 mutant non-small cell lung cancer, last presented by Pasi Yenny at ASCO 2024. This was the DESTINY-Lung02 study. As you can see, patients with metastatic HER2 mutant non-small cell lung cancer, incorporating most of the mutations we just reviewed, with more than one prior anti-cancer therapy, were treated with trastuzumab deruxtecan, either at the 5.4 milligram per kilogram or the slightly higher 6.4 milligram per kilogram dose. What we can see over on the right is that the response rates range from 50 to 56% for patients, depending on dose. Progression-free survival, 10 to 13 months, and overall survival not different between the two different dose arms, 19 versus 17 months. Based on this, the currently FDA-approved dose in HER2 mutant non-small cell lung cancer after one prior line of therapy is the 5.4 milligram per kilogram dose. Next, we'll move on to HER2 overexpression or immunohistochemistry positive non-small cell lung cancer and lung adenocarcinoma. There's a number of different articles demonstrating the positivity of HER2 expression in non-small cell lung cancer, maybe as low as 8%, as high as 30%, depending on the assay methodology. In addition, in the table below, it's demonstrated that there's many different ways that you can do scoring. There's a standard breast cancer scoring, gastric cancer scoring, and the more classical H-score. And there's fairly decent concordance, especially within the 2+ and 3+ realm between these different tests. So if you have a home-developed test against breast cancer or gastric cancer, that should be sufficient for using for detecting HER2 positive lung adenocarcinoma. Trastuzumab deruxtecan was also tested here in the DESTINY-Lung01 study. We can see that there were two different cohorts at the top of HER2 overexpressing non-small cell lung cancer at the two different dose levels, and patients were included with either 2+ or 3+ immunohistochemistry positive. When we look at the response rates, the response rate was lower than we see for HER2 mutant non-small cell lung cancer, 26% and 34%, depending on dose levels, actually slightly lower for the higher dose, with a duration of response around 6 months for both groups. Looking at the progression-free survival here, we can see that progression-free survival for the overall cohort of 5.4 milligrams was 6.7 months, overall survival around 11.2 months. What I thought was interesting about this is this HER2 overexpression seems to happen independently of many of the molecular alterations we find in lung cancer. So at the bottom, you can see that a few of the overexpressed tumors were EGFR mutant, many more actually had KRAS mutations, and there were other alterations too, including HER2 amplification as tested by FISH, which sometimes overlapped with overexpression, but sometimes did not. So I would encourage people to use the IHC test because there is an indication for trastuzumab deruxtecan for HER2 positive cancers, pan-cancer, and that includes lung cancer. We looked at other data here. So this was presented at World Conference in Lung Cancer 2024 by David Blanchard. And this study was interesting because it included more patients in part one of the TDXD monotherapy, more patients with EGFR mutant non-small cell lung cancer in a broader population that had HER2 overexpression. So when we look at the data here, very similar for the 5.4 milligram per kilogram dose is what we just saw in the last Destiny study. Progression-free survival around 8 months, median overall survival around 17 months. And some of the side effects that we'll get into on the next slide include many chemotherapy side effects from the topo-1 payload that's delivered from the antibody drug conjugate, including nausea, fatigue, anemia. Here they didn't see pneumonitis, but that's also something we'll go into in a minute. When we look at safety overall for trastuzumab deruxtecan, it's the same as in other indications. But one thing that's unique may be that pneumonitis has a slightly higher risk in non-small cell lung cancer. Total risk of pneumonitis might be as high as 15% across many of these studies. And this is adjudicated with centralized groups that looked and tried to sort out pneumonitis from disease progression, from just regular pneumonias that patients can get. So adjudicated pneumonitis was somewhere between 15 and even as high as 20%. There may be slightly higher risk for patients who had had immunotherapy within the last 3 months. Overall safety of the drugs, though, you can see on the far left, include many chemotherapy side effects, probably from the payload, common things that we're good at managing, and a low risk of stomatitis. I will note that alopecia is common with this, which is unique compared to most of our first-line treatments in lung cancer, which often include pemetrexed. So many patients have AEs, but these are felt to be fairly manageable, and this is an IV once every 3-week dosing and schedule. Moving on, there's exciting new data of HER2-directed tyrosine kinase inhibitors. So there have been many attempts to develop these, even drugs back in the day, such as afatinib, which is approved in EGFR mutant lung cancer, has some off-target activity in HER2 mutant non-small cell lung cancer, but seem modestly active. There are some TKIs that are active in breast cancer that are approved, but unfortunately, these are not quite as active against these exon 20 insertion lung cancer mutations. So in this study, the BEAMIAN Lung 1 study, a drug called zongertinib was tested at 2 different dose levels, 120 milligrams and 240 milligrams, with expansion at the 120 milligram dose in patients with tumors with a tyrosine kinase domain mutation, patients with the same mutation that were previously treated with a HER2-directed antibody drug conjugate, generally trastuzumab deruxtecan when it was available locally, and also patients with tumors with a non-tyrosine kinase domain mutation, such as that S310F. Over on the far right, I've summarized the data for you. In cohort 1, the response rate was 71%, disease control rate 96%, duration of response of those 71% of responders, 14 months, and progression-free survival exceeded a year, 12.4 months. Looking at the other two cohorts, the numbers were fairly small and they haven't reported the mature data yet. Response rates were somewhat more modest after previous treatment with a HER2-directed ADC and for the non-tyrosine kinase domain mutations. So for those other random mutations, only a 30% response rate, but still many patients responded. So I think we still have questions about exactly which of these mutations are most suitable for a drug like zongertinib. But I think these data, especially looking at cohort 1, I think back to the days of EGFR mutant lung cancer and the original data for gefitinib and erlotinib, and this response rate and progression-free survival is actually better than we saw for the original EGFR TKIs. So this is a solid targeted therapy, not yet FDA approved, not yet available, but certainly on the runway. Here's the progression-free survival curve showing over 12 months of progression-free survival, which is very exciting. And then there does look like there's some intracranial activity as well with these drugs. We know that all lung adenocarcinomas have a decent risk of brain metastases, especially those with targeted mutations. And when I think about HER2 mutant non-small cell lung cancer, I think much of the biology is fairly similar to classical EGFR mutant lung cancer or EGFR exon 20 insertion non-small cell lung cancer, which should not be confused with these exon 20 insertion mutations. But we do see some responses in patients with measurable brain metastases. 27 patients measurable, response rate 41%. Yes, this is lower than the overall response rate, but still demonstrating possible intracranial efficacy. And I do wonder about the different dose levels and whether the higher dose might have more intracranial efficacy at 240 milligrams. There's another TKI, this drug BAY for HER2 mutant lung cancer. This was a cohort of TKI and other trastuzumab-based therapy naive patients with lung cancer and HER2 insertion mutant disease. Demonstrated a response rate overall of around 70%, so partial response rate 70%, couple of complete responses on top of that, with the duration of response of 8.7 months. Progression-free survival, though, at 7.5 months, at least as of this report, numerically looks lower than that cohort one we saw from zongertinib, exceeding 12 months. Although I will note that most of the patients down here on the waterfall plot did seem to have some degree of response. So it's hard to do cross-trial comparisons, particularly between different phase 2 clinical trials with different selections conducted in different places. But I think both of these drugs look highly active and much more active than the drugs we've tried historically in as far as TKIs in this tumor type. What about safety and tolerability? Well, most of these have side effects that we expect from tyrosine kinase inhibitors in general, most notably diarrhea, rash, and some LFT abnormalities at the top of the list. Zongertinib, numerically, again, seems to have fewer overall side effects, with the BAY drug having 90% diarrhea going down from there. Zongertinib, only 50% of patients having diarrhea, especially at the 120 milligram dose. And then looking down to the bottom, fewer grade 3 events for zongertinib, fewer dose reductions for zongertinib. So again, I caution people to not think about cross-trial comparisons, but it looks like zongertinib may be more tolerable and potentially more effective. So in conclusion from this section, the antibody drug conjugates, especially trastuzumab deruxtecan, remains FDA approved for ERBB2 or HER2 mutant non-small cell lung cancer following prior therapy. We're thinking about its usage in the frontline setting, and that would be as a single agent, not in combination with other chemotherapy or with immunotherapy, but thinking about its use in the first line setting, but currently it's approved in the later line setting. We can think about HER2 positive non-small cell lung cancer with any other molecular alteration, and those patients being a candidate possibly for trastuzumab deruxtecan as well. And there is an FDA approved pan-tumor indication for that. And the HER2 TKIs are emerging. Very exciting data from just last week presented by John Haymack about zongertinib. Appears more effective and tolerable so far, and we'll see about other drugs that are emerging, because there are indeed many other emerging HER2 TKIs in development. They may have different safety profiles, tolerability profiles, and we'll keep an eye on CNS penetration too. Then I'm also hearing about HER2 bispecific antibodies and HER2 bispecific antibody drug conjugates. So many more tools in the pipeline that we can see in investigation in the future. Moving on to MET exon 14 mutations in non-small cell lung cancer. These are fun because for many, many years, we had looked for MET alterations in lung cancer, and we've been looking in the exons directly in the direct exon place. But these mutations are actually not in an exon. They're in the splice site just immediately prior to exon 14. So if you sequence whole exomes, you actually don't find them. You find splice mutations that drop out exon 14. And what this does is prevent degradation from MET from the cell surface, resulting in mass activation of MET. So it's basically the same as overexpression on the cell surface happening on a genetic level where MET is being produced but not degraded because of aberrant splicing. So it's worth keeping in mind that this is wild type MET for the most part. This degradation domain doesn't have much to do with the tyrosine kinase domain. So these are not really activating mutations in the tyrosine kinase. They're overexpression mutations. So capmatinib is currently approved for MET exon 14 non-small cell lung cancer based on the geometry study. In first-line therapy, it does seem to be somewhat more sensitive to in non-small cell lung cancer, with response rates pushing 70%, progression-free survival over a year, overall survival almost 2 years, as compared to in the same study when patients with prior treatment received capmatinib, response rate lower at 44%, progression-free survival half as much, 5.5 months, and overall survival numerically less. So even though chemotherapy and potentially immunotherapy can be active in MET exon 14 non-small cell lung cancer, particularly in patients with PD-L1 negative disease and not a smoking history, I would definitely recommend MET TKIs in the frontline setting based on potential more efficacy. Tolerability, peripheral edema tops the list, followed by nausea, vomiting, creatinine things, and LFT things. I would say in my experience, the dose-limiting toxicity is almost always peripheral edema. And I've had a number of patients that have to dose reduce to 3/4 dose or half dose. Emerging, Ben Creelan at Moffitt, as well as a couple of our patients, have put together a hypothesis and tried SGLT2 inhibitors. I don't have any data to share with you, but if anybody out there has any data or tried these, I think they can help with the MET-related edema, and really other things don't. So furosemide doesn't help. Compression stockings barely helps. Patients can't lie in bed with their feet above their heart for a certain amount of time. It's really kind of a puffy refractory edema that's really hard to treat. So if anybody out there tries the SGLT2 inhibitors, any of them are fine, let us know. Another type of MET alteration in lung cancer is MET overexpression. First, MET expression, protein expression at least, has been quite variable. And there's a number of different antibodies that have been tested, but MET expression in non-small cell lung cancer seems highly expressed when you just look by immunohistochemistry, and doesn't seem to be a strong predictor of response to therapies. So MET IHC, I think there's still some story about how do we define MET IHC there. On the other hand, MET amplification, so looking truly for multiple MET genes as compared to the CEP7, the centromere, and looking for a high ratio, or looking at next generation sequencing or potentially circulating tumor DNA, sometimes they can say there look like many more copies of MET than everything else in the DNA. So MET amplification does look like a driver. Telisotuzumab vedotin, or Teliso-V, looks active with overall response rates in the 35% for MET high, 23% for MET intermediate, and waterfall plots that look like there are responses in many, but not all patients. And when we look in this study, progression-free survivals in the 5 to 6 month range, and overall survivals in the 14-month range for this. So I think Teliso-V, after being evaluated by the FDA for a while, is being considered in this indication as an antibody drug conjugate that may be active here. When we look at side effects of this, in addition to the chemotherapy side effects, again, we see the peripheral edema, and then uniquely, probably because of the vedotin payload, peripheral sensory neuropathy that can be significant and sometimes persistent over time, even after stopping the drug. So in conclusion, for MET lung cancer, for MET exon 14 mutant non-small cell lung cancer, capmatinib and tepotinib are approved and preferred first-line therapies right now. There's a lot of other drugs that are MET-directed, including both antibody drug conjugates and tyrosine kinase inhibitors in development now. Next, we'll briefly touch on BRAF. So BRAF, there's 3 different classes of mutations. The most common in non-small cell lung cancer, and also the only one that's currently actionable, is the BRAF V600E mutation. The other classes are either kinase dead and don't signal through BRAF at all, or kinase active, but we don't have ways to target them right now. So all of these other BRAF mutations, when we see them, we're not exactly sure what to do besides maybe give standard treatments that we would give for otherwise mutation-negative non-small cell lung cancer. Encorafenib and binimetinib are emerging for this, both in previously treated and treatment-naive patients, with response rates in 75% for treatment-naive, 46% for previously treated, and good-looking progression-free survivals. Duration of progression-free survival looking around 30 months, actually, in the first-line setting, 9 months for previously treated. Keeping in mind that dabrafenib and trametinib are also approved in non-small cell lung cancer in the same indication, one of the advantages of encorafenib, binimetinib seems to be not seeing the fevers that we see that are sometimes associated with dabrafenib, as well as generally a slightly more favorable side effect profile. Overall survival had not been reached as of the previous presentation by Greg Riley at ESMO in 2024. But looks favorable. So encorafenib and binimetinib are FDA approved, reasonable alternative to dabrafenib, trametinib for lung cancer, also used in melanoma, and both are approved in that setting. And then the type 2 and the type 3 BRAF mutations currently aren't actionable, but options for these are in development and clinical trials. Finally, we'll move on to KRAS mutations in non-small cell lung cancer, first starting with KRAS G12C mutations, but then touching at the very end about brand new data and KRAS G12D mutations. So KRAS G12C mutations in lung cancer comprise around 40% of all KRAS mutations, are often smoking associated, and up until recently had not been actionable because KRAS can't be directly targeted in the GTP binding pocket. Instead, allosteric targeting methods need to be used. I'll point out that KRAS G12D is often less smoking associated and has the same challenges about targeting. Now, the G12C inhibitors took advantage of the cysteine residue. The cysteine residue allows covalent binding of small molecules and therefore targeting. So that was easier to target. And there are 2 drugs that have been used, adagrasib as well as sotorasib. So in adagrasib for KRAS G12C in the Crystal 12 study, overall response rates were 30% for adagrasib as compared to docetaxel, around 10%, which is how we expect it to perform, with waterfall plots showing response of the majority of patients after prior therapy. So this is following chemotherapy or immunotherapy or the combination. Looking again at progression-free survival 5.5 months for adagrasib, significantly better than docetaxel. But if we think back to what I said about drugs like zongertinib and the first-generation EGFR TKIs, we were seeing progression-free survivals of 11 to 12 months, seeing seeing response rates more in the 70%. So these drugs are definitely hitting the target, but not as highly active as some of the other small molecule targeted therapies that we have. There's quality of life analysis for this approved drug, though, and compared to docetaxel, quality of life does seem to be better. And these data presented at ASCO by Tony Mock with slower time to deterioration for adagrasib. We expect that for drugs that have a longer progression-free survival, but also more tolerability than docetaxel, especially when docetaxel is given in the 75 milligram per meter squared every 3-week dosing. Sotorasib from this publication in lung cancer also showed a more favorable side effect profile compared to the docetaxel, with side effects, as you can see, hotter and brighter colors, yellow, orange, red, being patients more bothered by the side effects of treatment. And you can see that persistently goes on from the beginning all the way through the far right as treatment continues on. Whereas sotorasib, patients are bothered by treatment more in the first half of the therapy, but then those side effects generally go away over time or become mild. So both of these drugs, adagrasib and sotorasib, look tolerable. Question, though, is without a lot of activity, can we combine them with other drugs in the first-line setting? Divarasib is another KRAS G12C inhibitor, but came later into emergence with a progression-free survival by itself in all patients of 13 to 18 months. Numerically looks pretty good. Response rates 55 to 59%. But can it be combined with immunotherapy? So in this second-line study, it was combined with atezolizumab. And indeed, response rates for no prior KRAS G12C inhibitors looked 55 to 61%. Many patients had shrinkage of tumor. Hard to interpret these data when we're comparing two therapies together, though, in a second-line setting of what we would expect in historical controls. But it looks like it can be tolerated and safety was okay. Olomorasib is also a G12C inhibitor, and in this cohort, including non-small cell lung cancer patients, it was actually boldly given in the frontline setting together with pembrolizumab, pemetrexed, and platinum. Now, this is only 21 patients, but response rates looked favorable. Of course, this is first-line untreated patients, so we expect them to have good responses. And when we think about side effects, we worry especially about LFT abnormalities, which had been a problem with immunotherapy plus sotorasib and, in part, adagrasib, which looked common but less common than with either of those other two drugs. Patients are getting anemia, patients are getting some LFT abnormalities, but mostly chemotherapy side effects. And I'll note that oftentimes these LFT abnormalities would get better when the olomorasib was held, as opposed to the immunotherapy held, suggesting that they were manageable, went away, and dose reductions could help. Moving on to the final and very exciting data presented by Casey Arbor at AACR 2025. This is a novel drug, zoldonrasib. First time I've publicly said that. Zoldonrasib for KRAS G12C non-small cell lung cancer. So zoldonrasib, again, like I said, KRAS can't be really targeted in the pocket. So they took advantage of a natural binding to another cellular occurring protein called cyclophilin A, which binds to a lot of things, and created a surface between cyclophilin A and the RAS-on stable conformation, which created a tricomplex between the small molecule and these three proteins in a covalent interaction that prevented RAS from signaling downstream. In zoldonrasib for KRAS G12D, the response rates overall were 61%, median time to response 1.4 months, and time on treatment only 2.6 months as of this very, very early presentation. But as we can see from the little arrows over on the left on the waterfall plot and the arrow heads over on the right on the swimmer's plot, most of the patients are still on treatment. So as these data mature, we'll expect to see these progression-free survival numbers and things like that hopefully be favorable in the small population of only 18 patients. Side effects from that seem tolerable. Usual TKI side effects: nausea, diarrhea, vomiting, AST, ALT increase, but all less than 50%. So look tolerable, at least across all of the patients, the 90 patients treated so far with the 1200 milligram dose, on whom safety have been reported. So in conclusion, adagrasib and sotorasib are approved for KRAS G12C. In my opinion, adagrasib looks modestly more effective and is slightly favored. Divarasib and olomorasib may be more compatible with immunotherapy and possible frontline combinations. And zoldonrasib is a novel KRAS G12D inhibitor with promising efficacy. There's a lot of other RAS-on inhibitors, including G12C RAS-on inhibitors, pan-RAS inhibitors, and other G12D inhibitors that are currently in development and in clinical trials that we'll keep an eye on. Thank you very much.